Enzymes have optimal environments.
3.2. Note, disclaimer.
In this video, we're going to compound what we learned in section 3.1. If you remember in section 3.1, we talked about some reactants and how we have some products, and that usually there is some energy that is required to go from reactant to product. However, with the addition of an enzyme, what ends up happening is we can lower that activation energy to go from reactant to product. An example of this would be something like water. So if we have H2O, that ends up breaking down into some hydrogens and some oxygens. Please note that this is not a chemical reaction per se that's balanced. I'm just showing you that you have some hydrogens and oxygens that break off. You don't need to know how to balance an equation for AP biology, and I understand that most students have taken chemistry prior and probably seeing a lonely hydrogen might be a little bit annoying, or lonely oxygen, but please understand, all I'm trying to say is that if you have some sort of reaction that exists, you're lowering that activation energy. You're lowering the amount of reactant that goes to product, and you're doing so by altering that enzyme to the reaction itself. So the act of adding the enzyme does that. Now, where does the environmental factor come in? Well, there are three main conditions that allow enzymes to work properly, and this includes temperature, pH, and substrate concentration, which we're going to dive into deeper in this video. However, I want you to think about your stomach, and you want to think about your stomach as being very acidic. That means that the pH is less than 7. And what happens in our stomach is we have certain enzymes that exist. And we want our stomach to be very acidic because there are certain enzymes that allow it to work properly in the stomach, as per se, maybe in your arm. So those are optimal pH related enzymes. So that's what we mean by optimal pH. An enzyme can work effectively for its purpose, its specific purpose, such as breaking down foods in the stomach if it's very acidic, if it's at an optimal pH. Same goes with temperature. There are specific temperatures within even the human body that certain enzymes work optimally in. If it's too high or too low, we say that there's something called denaturing, which we'll come to talk about later on. And we also have something called substrate concentration, which we touched upon in section 3.1. Sometimes you could have a reaction that's supersaturated, or that the velocity of maximum can be reached, which we're going to talk about a little bit in this video as well. But for now, what you need to understand is there are three main environmental factors that influence enzymes, which include optimal temperature, optimal pH, and substrate concentration. This is a great review of what we're going to be talking about in this video. I would suggest taking a screenshot of this because this table allows us to really hone in on the differences. For example, when we're talking about temperature, which is one of our factors, if it's low temperature, what happens is there could be reduced enzyme activity because you don't really have high kinetic energy, which means if you think about kinetic energy in chemistry, that means that if you have molecules and you have low temperature, they're not going to bounce as efficiently as you would think higher energy. You would think there would be more bouncing. So low temperature means that you have low energy, and that means that there's lower collisions. So if you want to think about it as a box, let's say that we have lower temperature, what ends up happening is those molecules are tightly packed. They're not really moving around as much. Whereas if you have higher temperature, you want to think of those molecules moving around, bouncing around, bumping into each other, sort of in chaos. So that's the difference between a lower temperature and a not so lower temperature. Now, let's take a look at the opposite extreme. If you have high temperature, that occurs, and then you have something called denaturing, which basically means that the 3D structure is now changed, which means that the functionality of that enzyme now changes as well. And this is really important to note because this can result in permanent loss of that enzyme structure and function. Usually it's irreversible in the 3D change. So the shape, once it contorts specifically, it can now not go back. And so it's deemed non-functional. And you can't have those molecules bind properly. So remember, you have a substrate that binds to an active site. So we have this substrate here, and usually it binds to the enzyme, and it binds at that active site. We call that induced fit. But say that this structure is now messed up. Essentially, now it won't be able to bind properly, it won't fit properly. That induced fit won't work. And so that's an issue with denaturing. And you can't return back to its original activity. This is same, so throughout when we talk about denaturing, so I want you to keep that in mind. Remember, we talked about pH level and acidity versus basic environments. If we have extreme pHs, what happens is that also leads to denaturing as well. So the shape can be altered. And then thirdly, we have salt concentration. Remember, we want to worry about that ionic structure. So going back to chemistry, so if you look at the periodic table, if ionic is if you have a plus and a minus, all the charges, if the interactions can't properly bind to each other with the pluses and the minus, so if you have some chlorine and you have some potassium ions and they're not able to interact properly, that means that it's going to affect that shape and functionality. So the common theme here is all of these factors have to work cohesively at the best condition. Otherwise, you're going to have issues with the structure and the function of that enzyme, which means it can't bind properly at that active site that we talked about. And because the structure is now altered and the substrate can't bind properly. So these are factors that affect enzyme activity and essentially alter the shape of that enzyme, which can be a problem.
Just a little warm air, please. Gotta look my best for catalysis.
You got it. Max heat mode, baby.
Wait, not that hot. My shape.
In that little video, you saw an enzyme and you saw that enzyme being affected by heat and eventually became denatured. So let's talk about it. So when we're talking about the shape of the enzyme, that really defines the functionality of that enzyme. And you might be thinking, what exactly does that mean? So in this little enzyme substrate complex that you see here, we have the enzyme and we have the substrate. And ideally, we know that at the active site, where the enzyme attaches to the substrate is important for turning the enzyme on. And this means that it needs to be induced fit. It needs to have a structure that allows the substrate to attach to the enzyme properly. However, enzymes are proteins, and what happens is when they are highly dependent on those shape, this can be influenced by their environment with things like heat, like we saw the blow dryer there, or pH or harsh chemicals. And essentially what happens is the enzyme will become what we call denatured. And denatured is not what we want to happen because what that means is now the enzyme shape and the active site where the substrate would attach is essentially now damaged. And therefore, that's what denaturing is. It's the influence of heat or pH or harsh chemicals to then alter the shape of that enzyme, therefore rendering it unusable for purposes that it needs to do. So when we talk about denaturing, it's not always a permanent process. Sometimes what can happen is you can have mild temperature changes, so slight heating that can allow for denaturing, but then eventually the enzyme can go back. Small pH pH shifts. For example, if you have an enzyme in the stomach and all of a sudden in the stomach it is not as acidic and then becomes a basic environment and then goes back down to acidic, sometimes that isn't permanently damaging the enzyme itself. And this also includes temporary chemical stress that can influence the behavior of that enzyme. For the rest of this video, or at least this third part, we're talking about inhibitors. And if you remember, one of the factors that we said earlier that could pose a problem when it comes to the behavior of enzymes is the concentration. Remember, we said concentration is really important. And so I want you to remember that when we're talking about inhibitors. Inhibitors are basically molecules that alter the behavior of this enzyme attaching to this substrate at the position that will allow the enzyme to turn on. Remember, you need a substrate to attach to the enzyme in that induced fit model to combine together to basically turn on this enzyme. If it's not able to attach, that basically means the enzyme doesn't work properly. So we're going to talk about these different factors that halt that process, and those are known as inhibitors. So if anything, you want to take a picture of this, I would do so, take a screenshot, because this locks in all the different types of inhibitors that you are expected to note. This is a very complicated table because I added some additional information in here, such as velocity and Km. Please note for AP Biology, you do not need to memorize this information right here. However, I did put this in here if you want to, because in the past, especially in the last 3 to 5 years, AP Biology has been providing a lot of tables and graphs, and there seems to be a lot of tests that are within the last 3 to 5 years that are utilizing tables and graphs. And with the updated curriculum, you never know when that's going to appear. There seems to be an uptick in the topic of enzymes. Because of this, I included these two factors in this table. While this is biochemistry, because I have a feeling, and I have noticed this in the past, there are tables and graphs related to enzymes, and your knowledge related to these two factors may be beneficial in understanding those tables and graphs when you are given them for an FRQ, for example. And we will explain this more in details in just a second, but for now, we're going to be focusing on the inhibitor types and understanding the binding site related issue and the effects on binding. We're going to later focus on these factors, and we're also going to give examples so you can help understand these concepts. Okay, so let's get started with competitive inhibitors. Remember, we said that the enzyme usually attaches to a perfect substrate. Now, let's say that the enzyme usually needs to attach to the substrate to allow your stomach to work effectively. Okay, and now let's say that we add a different item that is a competitive inhibitor, and call it C, that is competing for this specific enzyme. And instead of the substrate actually attaching, the competitive inhibitor attaches to this enzyme. What that means essentially is that this enzyme will not work because the appropriate substrate did not attach. Instead, the competitive inhibitor did attach. And competitive inhibitors are like two different cars that are racing for the same spot. So that's what we mean by competitive inhibitors. They're competing for that enzyme at that active site. Now let's talk about non-competitive inhibitors. So what ends up happening is, let's say you have an enzyme and you have a substrate. So the enzyme and substrate is here. And we have what's called an allosteric site, which is not considered the active site. So let's say that we call this position right here where the substrate would originally attach to the enzyme. the active site. And let's say that there is an item that is able to attach to a different spot on the enzyme, rendering it not working, meaning the enzyme not working. This is called the allosteric site. The allosteric site basically means that you have another molecule that is considered the non-competitive inhibitor that can attach to a different position on that enzyme, basically changing the shape of the enzyme. Remember, when the enzyme basically changes shape, that means that it changes the process of that enzyme. And so that's what a non-competitive inhibitor is. It's not going to attach to the enzyme at the active site. Instead, it's going to attach to a different position outside of the enzyme on a different spot called the allosteric site, which therefore renders the enzyme not working or not feasible to work. Let's talk about uncompetitive inhibitors. So that's the third one on this table. So what basically happens is you have a enzyme and you have some sort of substrate, and there's a position where something comes in called the ES complex, which is a location where there is a molecule that attaches and basically renders it unconscious or unable to work. It basically traps that substrate to the enzyme, to that active site, but basically there's something on the outside of that that attaches to it. And that's called the uncompetitive inhibitor. You could also have, which is the fourth one, called the mixed inhibitor, which could be a combination of different ones. Like you could have the allosteric site, or you could have the ES complex, which can attach together, and it affects both the binding process and how it lowers the activation energy. So you're not just having the binding site being an issue, but you also have the issue of how the enzyme fully is going to work in terms of catalysts behavior. We also have what's called irreversible, which basically means it's going to attach to the active site. But what ends up happening is it's permanent damage, which basically means that it destroys the enzyme. So you'll have the enzyme and you'll have this irreversible inhibitor that attaches. And then what happens is the enzyme doesn't even work once it attaches. So it's sort of like a competitive inhibitor, but not really. And so those are the 5 different types of inhibitors. Don't worry if it's confusing to you, because we're going to describe it in just a second. But before we get started, I'm going to show a little clip where you can understand the differences based on the different analogies provided. So competitive is racing for the same spot. We didn't talk about this, but non-competitor is when a manager cuts the power. Uncompetitive is when we trapping the worker and the tasks together. Mixed is chaos, basically everything's being affected. And irreversible is when the enzyme literally passes away and no longer can function.
Almost there.
Not today, buddy.
Bro, that was my active site.
Should've parked faster. Let's get this reaction poppin'. Wait, why is everything dimming?
Boss, you okay?
Just flipped your off switch. All right, let's make some product.
Wait, did it just?
We stuck?
Oh yeah, together, forever.
Let's talk more into details about the different types of inhibitors. Warning or disclaimer for AP Biology: over the course of the years, there have been more prompts or essays related to enzymes. And while all the information provided here might be a tad bit, a little bit extra, more than the actual exam, the reason why I'm providing this information is so that way you know how to interpret graphs and tables when it comes to enzymes in a prompt. So first, let's tackle competitive inhibitors. So competitive inhibitors, if you notice in that little video, you have 2 different blobs or molecules that are competing for the same parking spot. And that's how competitive enzymes work. Basically, the enzyme has to attach to the substrate essentially at the active site. What happens is you want the enzyme to attach to the substrate in a perfect reality. However, with competitive inhibitors, they're fighting for that same spot. And essentially what happens when the competitive inhibitor actually attaches, that means that the substrate can't turn the enzyme on. So what happens essentially is it occupies that position, and the affinity for the enzyme and substrate becomes stronger. So you need or want more substrates to be able to compete with that competitive inhibitor, essentially. So that is what a competitive inhibitor is in terms of enzymes. Then you have what's called a non-competitive inhibitor. You want to be thinking about that little clip where we said, oh, well, there is a molecule that essentially turns off the enzyme. And that sort of works the same way. Yes, you have the active site, but you have a different site called the allosteric site. So this is the allosteric site here, and this is the allosteric site here. Both examples of non-competitive inhibitors. And in this case, what happens, regardless of whether or not there is a substrate attaching or not, the allosteric spot is what occupies the non-competitive inhibitor. And once it does that, it essentially turns off the act of the substrate attaching to the enzyme at the active site. So it doesn't matter if it's attached or not. By it, I mean the substrate. If the allosteric site is occupied by the non-competitive inhibitor. Essentially, it shuts it off. And so what ends up happening is the velocity at which the enzyme can work will actually slow down. So it will reduce, and there is no change to the affinity between the enzyme and the substrate. It's just that the non-competitive inhibitor, regardless, will shut this off, whether or not there's a substrate or not. And so that's what a non-competitive inhibitor is. Lastly, we have the uncompetitive inhibitor. By lastly, remember there are two more additional examples called mixed and irreversible, which we'll cover in just a second, but these three are the most popular ones for exams. So I wanted to cover these in a whole package scenario. So when we talk about this, or essentially when we talk about uncompetitive inhibitors, you want to think about the fact that yes, you have the substrate that attaches to the enzyme. Well, what happens if it basically traps that substrate forever? So what happens is the uncompetitive inhibitor will trap the substrate to the enzyme, just indefinitely keeping it on and working at all costs. So what ends up happening is you're reducing the enzyme substrate complex affinity, or the Km. That's what we call Km. Because you have that substrate, you attach to the enzyme, it turns on. There's no additional need needed because once it's locked, it's locked. And the velocity or the maximum is also reduced as well, the maximum potential, because once it's locked, it's locked. So that is a uncompetitive inhibitor. Once again, these terms of V max and Km are additional foundational terms that we're talking about just so you have an additional knowledge about the relationship between the two, because we are now diving into these graphs. These are potential graphs that you could see on an FRQ to compare the relationship between competitive, uncompetitive. Remember, mixed is the one we didn't talk about, and non-competitive. Mixed is a combination of both. So remember, mixed is when we have effective binding and catalysts. So this is at the allosteric site, and then what ends up happening here is the inhibited and uninhibited when you're taking a look at the relationship is you have the velocity, which is the speed, is going to go ahead and essentially decrease. And then what ends up happening is the speed, or the Km, or the affinity, varies because it's mixed, right? It's going to be mixed. The slope changes and the v-intercept changes essentially, but you can see the uninhibited one is going to be underneath and is going to be at a certain slope. whereas the slope of the mixed is more steep. So that's something to note. So it's a mixed behavior. It's not going to be straight through when we're talking about that relationship. Now, if you take a look at non-competitive inhibitors, which we talked about before, remember non-competitive inhibitors, if we take a look at the graph here, is when you have the allosteric site and it's attaching at that spot. So regardless of what happens, as long as the allosteric site is occupied, the substrate attaching to the enzyme doesn't matter. So keep that in mind, because when we're talking about non-competitive inhibition here, if we are inhibited, what that basically means is we're saying that there's going to be changes in both the slope and the y-intercept and the velocity at which it reaches its peak for the inhibited is essentially going to decrease. So we have it decreased. So that means it's going to be more closer to the uninhibited. It's going to essentially be that we're not having much speed, because once it's occupied and it has no competition, right? So if we have the allosteric site, it turns it off, There's no speed in which it goes quicker and quicker and quicker to produce. So I know that seems kind of confusing, but it's just to understand that once you have the inhibited, it's going to sort of taper off a little bit, because once you're binded, no substrate attaching to that active site is going to matter, because you have the allosteric site being occupied. So once it's occupied, it's occupied. Now, remember we talked about uncompetitive inhibitors. So uncompetitive inhibitors are that permanency on factor. So it's sort of trapped in that motion. So you'll see that the slopes look very similar. They're very much the same. And what ends up happening is, because it's trapped, you don't have any peak that it needs to get to. It's just forever on. That enzyme is forever on, turned on by the locked, not lock and key, but locked enzyme that's attached to the substrate due to the uncompetitive inhibition. And it locks that substrate in, decreasing the need for affinity, because once it's affinity, and decreasing the need for the velocity to maximize. So that's the difference. And then you have competitive inhibitors, which we talked about, is comparing the fact that we have an enzyme, we have the active site, and we're competing for that one spot. And that competition means that the inhibited is going to have no maximum velocity, and then there's going to be a greater increase in the affinity between the enzyme and substrate. So there is no change in the velocity itself, the speed at which it goes, but there is going to be an increase in the need to attach, because we're competing for that one spot. Then what isn't shown here is the irreversible, because really there's nothing that exists. It's just a zero graph. There's no maximum, and the enzyme is just inactivated, or nothing really happens. So with mixed, you get a combination of both. Irreversible, you get a complete altered stop. So that's the difference between the two. This relationship graph page seems kind of complicated. Just want to preface this by saying you do not need to memorize this. This is just to show you the relationships between the two. So if you are given a question where you have a graph and you're looking at it and kind of interpreting it and getting confused, you can sort of utilize your knowledge here to deduce that, okay, is this a competitive inhibitor? Is this an uncompetitive inhibitor? Mixed inhibition, pure non-competitive inhibitor, especially if they're giving you some sort of drug or some sort of treatment on a patient, and they're letting you know that, hey, there's an enzyme here, but it's not working properly. You know what the graph is supposed to look like, or your data is supposed to look like if they gave you a table, what essentially going to look like. Now, do you need that information for the exam? No, because you should be able to look at it and sort of understand what it's supposed to look like based on the information that's given. But I think now that the AP biology exam is getting harder, I thought this would be a nice additional way to help you out when reviewing those tables or graphs.
Everything's running smooth today.
WRONG, you're too slow and you're holding that substrate wrong.
Yo, chill!
I don't chill, I chaos manage.
VMAX, dropping.
One sip and you'll never be the same. Mmm, tangy. Can't move. Permanent bond, baby.
Now let's take our knowledge up one step, comparing competitive inhibitors to non-competitive inhibitors and uninhibited when it comes to enzyme substrate complex. So to recap, if I'm talking about uninhibited, I'm just talking about a normal enzyme and I'm attaching it to the substrate at the active site, and that's what we usually have. But if we have a competitive inhibitor, remember we have the enzyme, we have the substrate, but it's competing with this sort of additional substance called the competitive inhibitor for that active site. And then with non-competitive inhibitors, we have the enzyme, we have the substrate, which wants to attach, but at the allosteric site, which is a different spot on the enzyme, it can attach. By that I mean the non-competitive inhibitor, basically shutting off the enzyme and substrate complexity. So let's start talking about the reaction itself and how it works. If we have an uninhibited reaction, which normally is just the enzyme attaching to the substrate, this is what we want, where we have the rate of reaction. It goes through once it reaches the velocity, the maximum velocity, and the reaction has occurred. And then what happens is it reaches a maximum velocity. So in a normal case scenario where we're just having a normal reaction, we have a low substrate, slow reaction, fewer enzymes, and then substrate collisions, I should say. And then we have an increase in the substrate, which means that we start speeding up the reaction rate, and the reactant going through to be the product with the action of the enzyme attaching to the substrate will eventually peak. And this means that we've reached a saturation point where we just have the maximum amount of enzymes and the maximum amount of substrates that can attach, which means that it went through reaction to product and we have received or attained the maximum velocity. That is the ideal situation. So this curve right here is when conditions are perfect. You have enzymes, you have substrates, there's nothing bothering it, and you are uninhibited. You go from reactant to product and you reach that maximum velocity. Now with a competitive inhibitor, what happens is you have this sort of tapering that wants to get there, but what ends up happening is you're competing for that spot because you not only have the substrate itself, but you have this other competitive inhibitor. So it sort of is going to compete for that spot and start going up, but not working as best as uninhibited because you have that competitive inhibitor blocking that spot. And eventually, if the competitive inhibitor attaches or the substrate attaches, it's going to peak, but peak a lot. It takes not a great steep. It just goes a lot quicker to get to the Vmax. So the rate of reaction doesn't go as swift. Now, the least swift is the non-competitive inhibitor, because remember, we have the enzyme, we have the substrate, and we have the non-competitive inhibitor. So once the non-competitive inhibitor attaches, stops. straight right there. It doesn't matter how many substrates are there. And so it's going to stop a lot quicker to receive that V max. So essentially what this means is that if we have too little substrate, we have slower reactions. If we have too much product, we slow down from what we call feedback inhibition, which basically means we have other processes, which you'll talk about in cell communication unit, where other cells will communicate to each other to tell, okay, we have this enzyme, it's producing too much product, we need to slow down sending the substrate to attach to the enzyme. So that is when you have too much product. And product concentration means that if we have a high product level, meaning that we have a lot of enzymes being attached to a substrate, means that we have slow feedback inhibition. So we have to slow down that process, and this eventually decreases the activity. So it's not only the concentration of the inhibitors that impacts it, but there are outside factors that concentration of enzyme activity can work upon. So for example, you're going to talk about this in a later unit, but you could have a cell here that notices this cell has a lot of enzyme attaching to a substrate, and it will send messages, which is called a feedback inhibition. So it sends feedback to this cell to basically say, you don't need any more substrate, and essentially will send fewer substrate over so it doesn't continue to attach to an enzyme. And that's when you have that velocity reach its peak a lot steeper. So that's a perfect condition when it comes to that in general. So I hope that this in general shows you the relationship and how the rate of reaction can be affected by the concentration. Now let's talk about the final factor, which is temperature. So if we have high temperatures, that usually means that we have faster reactions because we have more collisions that occur. Each enzyme has its own optimal temperature, which basically means it works specifically. The enzyme works at its maximum efficiency. If it's too hot, we have denaturing, which means that it changes shape, and essentially that reaction can stop. So essentially we want temperature to boost that enzyme activity, but only up to a significant point. If we have too much heat, then what ends up happening is we have denaturing. So we don't want that to happen. Okay, now that we're done reviewing that, let's try to start a quiz question. Take a moment, pause your video, and see if you can answer this question. Okay, now that you're done reviewing the question, let's see if you got the correct answer. So in this question, it says, in a renewable energy research project, science are testing an enzyme that catalyzes the breakdown of plant material for biofuel production. When substance Z is introduced, the introduction of the production of the biofuel completely stop. Even if we add more material, when the substance Z is removed, the reaction immediately resumes to full efficiency. So they want to know what kind of substance is substance Z. Is it a competitive inhibitor, non-competitive inhibitor, cofactor, which we didn't talk about, an allosteric enzyme? And the correct answer is a non-competitive inhibitor. And the reason why you can tell this is if it's a competitive inhibitor, it's going to compete for the same spot, and rendering that it's going to compete, but it's still going to have some sort of effect. In this case, they tell you the production completely stops. And with a non-competitive inhibitor, remember, a non-competitive inhibitor means you have an enzyme, you have a substrate, and then you have this allosteric site in which you have the non-competitive inhibitor touches. So once it attaches to the enzyme, completely stops. The enzyme no longer is valid, and that's the key here. It says it stops completely. And so that's how you know it's a non-competitive inhibitor. And competitive inhibitor is still going to produce some sort of biofuel. However, it's going to produce at a slower pace. So if you see it slows down, then that's your key indicator that it could be a competitive inhibitor. I hope that helps.
Appreciate you riding along for our cosmic journey. If today's mission brought you the knowledge that slaps, make sure to hit that like button to boost that signal. Subscribe if you're locked in for our next Watch. And don't miss the link tree. It's got all the goods for AP Bio, PSAT Math, and SAT Math. Before I sign out, make sure you're all in one orbit and diving in. Killing out. Till next time.